Just for introduction, meroplankton is a wide variety of planktonic organisms which spend a portion of their lives in the benthic region of the ocean. Anabas is a genus of climbing gouramis native to southern and eastern Asia. In the wild, Anabas species grow to long. They inhabit both freshwater and brackish water for an extended period of time. This is the taxonomy for the Anabas species. Environmental factors are often strongly affected reproductive success. In this respect, salinity is a key factor influencing the reproduction rate in aquatic animals. We used Anabas testudineus, a climbing perch fish, to experimentally assess the effect of salinity of the bird nest water. Apart from testing Anabas testudineus with bird nest water for its salinity, we decided to use it to find out if it speeds up the cell growth since it contains 5.27% of proline, which is said to promote cell generation, according to HelloNest 2015. The objective of this study.
Moving on to the methodology part, which consists of obtaining the eggs, preparing the solutions, and observing the age stage. The freshwater fish, Anabas testudineus, was used in this experiment. The sex of the fish was determined by observing the genital papillae, color, and body shape of the fish. First, weight the fish using analytical balance to determine the amount of hormone needed to be injected into the fish. The following formula was used to determine the amount of hormone needed for the fish according to its sex. Next, the fish was injected with overprim, a hormone used to induce spawning. The fish was injected at the part of the dorsal fin below the fish scale to make sure that the hormone penetrates into the flesh. Then, remove the needle carefully to avoid the hormone from leaking out. After injection, the fish were transferred into an aquarium and kept in a dark condition to allow spawning to occur naturally. The aquarium was observed from time to time to record the time of spawning. Next, preparation of the solution. 2 beakers of 250 ml were prepared for both dechlorinated water and bird nest water. The bird nest water was diluted by using only 12.5 ml and mixed with distilled water until it reached the total volume of 250 ml. The concentration of the experimental solution was determined by using the following formula. The parameters such as salinity and pH of both control and experimental solution was determined using refractometer, pH meter, and hydrolab quanta. After 5 to 6 hours, the eggs were obtained by scooping the eggs in the aquarium using a beaker. The eggs were transferred into the control and experimental condition. Only the transparent and floating eggs were chosen. A few drops of the eggs from the solution is added to a diffraction slide and covered with cover slip. Excess water was removed using a tissue. The slide was observed under the microscope. The egg stage were observed from one cell until it hatched for 24 hours.
Okay, as for the results parts, this is the initial parameter and final parameter for our tank. The fertilized time is 7:04 p.m. which is about 90 minutes late compared to tank A, B, and D, which is 6:45 p.m. This will be discussed in the discussion later. Okay, we have two test solution, which is the control and experimental. Moving on to the stages. Firstly is the zygote stages, which is one cell. We could not retrieve the pictures for experimental due to we couldn't find the stages at that moment. As you can see, this is the transparent line here is the chorion. The white dots here is the oil globule, and the space is the perivitellin space, and this is the egg yolk. And during the cleavage period, 4 cells, the blastomeres are more distinct compared to 2 cells. This is 8 cells, 16 cells, and 32 cells. How to count it? If you can see here, there's 1, 2, 3, 4, which is a 2 times 4 array of blastomeres. The same goes with 32 cells, 64 cells, 128 cells, and so on. The blastula period starts from 120 cells, which is the distinctly form of blastoderms, the 512 cells, 1K cells, and height. To differentiate between the height stage and 1K cells, you can clearly see the YSL nuclei here. The oblong stage, it shows an elliptical shape, while the sphere it shows a spherical shape. During the dome, we can see the yolk starts to bulge toward the animal bowl. Then this is 30% eboli. Gastrula period, which is the 50% eboli, you can see that blastoderms remain uniform thickness here. Moving on to the germ ring, from the upper view you can see that it forms a ring. While this is the lateral view, this is the shield, and then this is the 75% eboli. You can see that the dorsal side is distinctly thickening compared to the others. But stage, you can see clearly the notochord rudiment, and then the tail bud prominent. You can see that there is a slight curve here. Segmentation period, as for 4 to 5 somites, you can clearly see the Kupfer's vesicle. 8, 9, and 12 somites, we can simply count the somites. Here in somite stage, we can see that the brain neuromer starts to form slowly. In somites, the somite are already V-shaped, and there's 18 somites here. Okay, during the 21 till 23 somites, the tail is well extended, the brain neuromus is more prominent, and there might be a slight muscular twitch. Alright, so moving on to the pharyngular period, which starts off with prim 5. The brain neuromers are more prominent, the retina and skin is more pigmented compared to 4, and the tail is more extended compared to 21 till 23 somites. You can also see that there is a heartbeat and blood circulation in this stage. Okay, moving on to prim 15, the brain neuromers is more prominent, the retina and skin is more pigmented compared to prim 5, and the tail is much more outward and straight compared to prim 5. We can also see the heartbeat and blood circulation clearly in this stage. Alright, however, for control, the growth is suppressed. See that in the experimental beaker, the eggs hatch to a fry. In the control beaker, we are quite lucky enough to see there's only 1 or 2 eggs that hatch into a fry. However, this fry looks deformed. Okay, I will show you in this video. Alright, as you can see from the video, the fry is already out of its chorion, and at this moment it slowly sucks the egg yolk into its body, which is a pretty bizarre way for it to hatch compared to a normal hatchling. And the look of this fry itself is a bit deformed compared to the fry in the experimental. And this is the egg yolk from a dead or burst cells. As for discussion, I will do a comparison of the growth development of the eggs between the control and experimental by looking at the hour post-fertilization against stages of the cells. Okay, at an early stage, we can see that the eggs in the experimental beaker show a faster growth rate compared to the one in the control. However, at this point, which started off at 11:53 p.m., there has been a fluctuation of growth development until the end between the control and the experimental. As you can see that the growth development outcompete each other alternately, and this is because there has been a fluctuation of temperature ranging from 23 until 30 degrees Celsius. However, at the end we can see that the eggs in the experimental hatch faster, which is at 4:33 p.m. and the eggs in control hatch at 6 p.m. and show a deformed characteristic. In the experimental beaker, the primitive still show a positive result. However, in control, the growth of the eggs is suppressed. This is because most of the eggs are either dead or burst, and some show these stages even though we wait until 6 p.m.
During the experiment, the temperature recorded ranged from 21.5 to 30 degrees Celsius. During this time, there was a fluctuate in the growth due to the change of the temperature. The change of temperature is due to the alternately turning off the air conditioners, which affect the development of the eggs. Based on subscale Sing and Desai study, temperature at 26 degrees Celsius is the best for the eggs to hatch. Next, based on a study conducted by Nadira et al., the study demonstrated that water salinity at pre-PPT recorded the highest percentage of hatching success and yolk sac larva survival. The study suggests that a slightly saline environment may increase hatching success and yolk sac larva survival. Thus, the salinity condition for both control and experimental condition is ideal for the development of the fish eggs.
The aeration tube in tank C was slightly broken and caused the fish to stress out. This could affect the rate of cell growth. In this experiment, pH did not do much effect. The cell division for fish eggs in the bird nest water was faster than fish eggs in the chlorinated water in this experiment because bird nest water contains nutrient which speeds up the cell growth. According to HaloNest 2015, bird nest contains 5.27% of proline, which helps in cell generation. Furthermore, it also contains EGF-like that stimulates tissue growth and generation. Chlorinated water also has zero nutrient, which makes the growth of the cell slower than cell growth in the bird nest water. This condition makes the eggs to eat their own yolk to gain nutrient.
Okay, for the error, some of the error that we encountered during the experiment is first, during the pre-fertilization period, food tank believed to have stress condition. One of the factor is because there is no aeration in the aquarium. The fish is jumping around in the water, which is also one of the signs that indicate the fish is in stress condition. Okay, next, during the observation of the fish egg, the light of the microscope caused our fish to dry up, and this caused our fish to crack. This is one of the example of our fish egg that crack due to dry up.
In conclusion, it is clear now that the salinity of water play an important role in the survival and the development of the fish egg. According to the Moloku and Opowasili 2002 as cited in Musa et al. 2014, salinity is one of the factors that contribute to the hatching success of freshwater fish. Therefore, between our two parameters, which are bird nest water and dechlorinated water as a control, the bird nest water show a more successful development and hatching compared to the control.